So I would start first thanking the organizing committee for putting together this really amazing conference and of course for giving us the possibility to present our work today. So going directly on the topic of my talk, I will today talk about cardiac fibrosis, which is a maladaptive remodeling of the myocardium happening after damage of the heart, such as myocardial infarction. And actually, cardiac fibrosis is hallmarked by contraction impairment and excessive extracellular matrix deposition. And if we look at it from a cellular point of view, surely cardiac fibroblasts are the main player in these disorders. And what happens during cardiac fibrosis and what we can observe is a proliferation of this cell population, a transition and change in phenotype from fibroblast to myofibroblast, followed by an extensive ECM deposition, and finally an ECM stiffening. And let's say the main key driver of cardiac fibrosis are for sure profibrotic biochemical stimulation such as the TGF-beta, but mechanical stress have been identified as one of the main drivers of this pathology. And for this reason, our focus was to generate and try to generate a model that could replicate the mechanical alteration in this pathology. And for doing so, we developed a miniaturized platform which is able to apply control and tailor mechanical stimulation to three-dimensional construct. And you can appreciate it, a sketch of this device. From a top view, you can see that what we actually do is to see the three-dimensional cell-laden hydrogel in a central channel of a microfluidic device, and we can do it thanks to the presence of two rows of posts which confine our construct. But if you see the cross-section of the device, actually the posts are not pass-through, but there is a gap underneath. You may already have seen it in the talk of my colleague before, and thanks to this gap and controlling actually the geometry of the post, we are now able to transform a movement of the membrane in a precise mechanical stimulation felt by the cell in 3D. As in this case, you can see from the video, for example, cells feel a uniaxial strain. And this technology called uBit is today integrated in a commercial platform called uBit Platform, which is developed and commercialized by Biomimex. And here I have to disclose that I also serve as CEO and I share equity in the company. And actually, in Biomimex, we are using this platform for generate different models which all have as a common denominator the importance of mechanical stimulation. In particular, we can, for example, differentiate and maturate a healthy model of human tissue. You may have seen the poster of my colleague, Dr. Roberta Visone, about our uHeart model to test cardiotoxicity. We are also working, for example, on a gut model. But more importantly for this session, we also are generating a pathological and disease model in which we use and we induce actually mechanically related pathologies. And again, we just had the talk of my colleague Dr. Rasponi in previous session about osteoarthritis. And in a similar fashion, I will show you now how we apply this concept to cardiac fibrosis. So to start with, we start collaborating with the University Hospital of Basel, and we wanted to test the hypothesis that mechanical stresses, even in the absence of profibrotic biochemical stimulation, are actually enough to act as a key player in activating cardiac fibroblasts, eventually promoting cardiac fibrosis. And to start with, we work with a mouse model first, so we seeded actually neonatal rat cardiac fibroblasts in the platform, and we compared the effect of mechanical stimulation with the one of the more classical TGF-beta1 profibrotic stimuli. And actually the mechanical stimulation we choose and tailor in this case is one resembling the heart beating, so a uniaxial strain with 1 Hz of frequency and 10% of intensity. And what we could see, so we follow all the different steps of the pathology, we could first see that in a similar fashion to TGF-beta1 stimulation, also our mechanic stimulation could induce and enhance fibroblast proliferation first, but also a switch in phenotype from fibroblast to myofibroblast. And as you can appreciate from this immunofluorescence, with TGF-beta1, cells start to express alpha-SMA, which is a key marker of myofibroblast, but this is even enhanced in the presence of mechanical stimulation, even if TGF-beta1 is not in the model anymore. We went on and we checked how this affects ECM deposition, and again, as expected, TGF-beta1 enhanced deposition of proteins such as collagen 1, fibronectin, and aggrecan, but mechanical stimulation did the same, even in a more strong and evident fashion, again, even in the absence of profibrotic stimuli. And this results in the end also in a stiffening of the matrix that we could analyze by atomic force microscope and in a deposition of a more uniform matrix in the presence of mechanical stimulation. So now, to complicate a bit the model, we also went on and asked ourselves what happens if we introduce another important cell population found in the heart, so cardiomyocytes, and can the presence of functional cardiomyocytes somehow reduce the percentage of activated fibroblasts and so prevent the formation of a fibrotic setup. And still in a neonatal rat model, we now investigated the effect of different ratios of cardiomyocytes and cardiac fibroblasts when applying mechanical stimulation. And we did a lot of study on this. I will just show you some data, but basically what we could understand is that there is a ratio in the proportion of fibroblasts and cardiomyocytes, and below, for example, 50% of presence of cardiomyocytes, the fibrotic event cannot be stopped anymore. So when we have more cardiomyocytes in the model, of course the model is more protective against fibrosis, but when we go down, that's not the case anymore, and also from a functional point of view. So under a certain threshold of cardiomyocytes, actually the mechanical stimulation impairs even the contraction, and even by pacing the tissue, we cannot get any functional activity. So all of this was, of course, really interesting, but was done with animal cells. And of course, we all know that human models are what is relevant in this field. So we started to translate the finding and using, in this case, human atrial cardiac fibroblasts, generating our uScar, which is a human model of cardiac fibrosis on chip. And the setup at the beginning was exactly the same I showed you before. We just used a human cell type, and again we confirmed that by applying mechanical stimulation we can actually induce a triggering of fibroblast to myofibroblast transition, in this case even in a statistically significant way as compared to only TGF-beta. And again, we could confirm that our mechanical stimulation is enough to induce a fibrotic deposition of collagen 1, aggrecan, and fibronectin proteins. So now we were ready to validate the sensitivity of our uScar model against antifibrotic preventive drug. And here I show, for example, the work we have done with tranilast, which is an anti-allergic agent also used in the treatment of inflammatory disease and fibrosis. So actually we wanted to test the preventive effect of this drug, so we applied it in the system from the beginning together with dynamic stimulation. And what we could observe is that when tranilast is present in the model, actually counteracts the transition of fibroblasts to myofibroblasts, so it stops somehow the onset of cardiac fibrosis, and this is also confirmed by the fact that we can avoid excessive deposition of its extracellular matrix, again with a reduction in collagen type 1, fibronectin, and aggrecan deposition in the model. So we now have a functional and validated model of cardiac fibrosis on chip, and we wanted to start using it to test some innovative therapies. And in collaboration with Politecnico di Torino, we started to investigate the effect of an advanced therapy that they are developing there to actually reprogram fibroblasts into cardiomyocytes. So our colleagues in Politecnico are modulating the microRNA expression, so they are using a combination of microRNA to actually reprogram cardiac fibroblasts into cardiomyocytes-like cells, and this will be think about a strategy for myocardial regeneration after ischemic heart disease. And they did a lot of nice work that is published in a simple 2D model showing that when reprogramming cardiac fibroblasts with this MIR combo, so combination of microRNA, they could actually observe an enhancement in cardiomyocytes-related genes such as troponin type T and troponin type I, and a reduction in a statistically significant way of marker of fibroblast, showing that they are actually able to reprogram cells. And they also confirm it by FACS analysis showing that at least 10% of their population express cardiac troponin in the model after MIR combos administration. So of course this work was really promising, but they started to interrogate themselves and ask what happens if we put the same therapy in a more clinically relevant setup. And of course, talking about the heart, the main clinical relevant parameter is mechanical stimulation. And for this reason, we started to check together what happened with the advanced therapy in our system. And first, we decided to check how mechanical stimulation affected the efficacy of transfection. So cells were transfected in 2D, seeded in our model, and we compared the results when we apply or not mechanical stimulation. So starting from the static results, what we could see is that transfected cells seeded in 3D in our model actually start to reprogram themselves. So Mir combo is effective in 3D. Again, cells express a higher level of cardiac troponin T and myosin light chain. And as you see from immunofluorescence, they also express alpha-sarcomeric actinin, a key marker of cardiomyocytes. So far, all good. What happened then if we apply mechanical stimulation, which is actually what it will be faced in vivo? Well, results at the beginning were a bit surprising, but actually what we could see is that when we apply mechanical stimulation, efficacy of this advanced therapy is not clear anymore. So upon mechanical stimulation, actually cells do not express marker of cardiomyocytes, do not express alpha-sarcomeric actinin, while on the other side, they start to express higher level of collagen type 1, and collagen type 1 started to be deposited in the model, meaning that this is a first warning for this therapy, so telling us that mechanically active environment seems to hamper Mir combo efficacy. And we went a step further, and we also asked ourselves what happens now if we apply Mir combo directly on the cardiac fibrotic model, which is in the end the final application for this therapy that will be applied in a pathological model of heart. And once again, what we did was to generate first the model, then in this case to apply Mir combo in the chip and check what happened. Here the results were similar to the one I showed you before and confirmed that in the presence not even only of mechanical stimulation but really of a pathological environment, the efficacy of this therapy is not there anymore. So cells do not reprogram. Actually, they start to express even more marker of fibroblast-like cells. And this, of course, comes with a second warning. So this therapy is not enough to counteract cardiac fibrosis. One of the reasons maybe that is not effective on pathological fibroblasts. And so what we are thinking now and what these results allow us to do is think about a combinatorial strategy, so potentially applying this therapy together with an anti-inflammatory or anti-fibrotic compound. And with this, I would like to conclude just with a short recap of what I told you today. So we generated a model called uScar, which is able to recapitulate all the phases of cardiac fibrosis, and this only by applying a mechanical stimulation. So no need for biochemical factors, and this, of course, is an advantage because we can avoid crosstalk with antifibrotic target. Our model allowed to study the crosstalk between different cell populations in the heart, allowed to test efficacy of drug, but most importantly, allow us to better study new and advanced therapies. So thanks to our model, which is really a complex model, we could elucidate limitation of an innovative therapy that were not clear in 2D, but not even in a three-dimensional complex model. And so I would just like to give you as a take-home message the fact that recapitulation of all the relevant traits of a pathology, and in this case, for example, the alteration of mechanical environment, is really fundamental, especially when we want to generate clinically relevant model of pathologies. So 3D and miniaturization may not always be enough, but we really have to think about all the different components that the pathology is facing in vivo. And with this, I would like to thank all the people working in this project, starting, of course, from my colleague from Mimic Lab in Politecnico di Milano, especially Professor Rasponi and Roberta Visone, which performed most of the experiment I show you, my amazing team at Biomimex, and all the collaborators and founding agency to allow us to work in this project. And thank you for your attention.